Okay, great. So, everybody, welcome to our Journal Club with CANN. I'm Twinkle, the head of publications for CANN right now, and I'll be hosting Journal Club for the rest of the year. And joining us, we have Dr. Anna Schwab, an expert in cannabis science and education. She holds a BS in cellular and molecular biology, an MS in biological sciences with the focus on plant population genetics, and a PhD in biological education and cannabis genetics. Her award winning doctoral research, a multifaceted approach to investigate genetic aspects of cannabis, has resulted in multiple journal publications. Currently an associate professor at the University of Colorado, Dr. Schwabe co-teaches modern cannabis science currently in its fourth year. She is the director of research and development at Shore/420 Organics in New Jersey, where she also spearheads a comprehensive cannabis education program in collaboration with the Agricultural Genomics Foundation. A distinguished presenter and published researcher, she is a prominent figure in the cannabis industry, and that is why we're so pleased for her to join us this week on Journal Club and share some of her research with us. So thank you so much, Dr. Schwabe.
Oh my gosh, thank you so much for having me. I'm delighted. And this research I haven't talked about in literally years. So I'm so excited to actually present this. And I also haven't so if you've read the paper, I'm going to present this in a different way. So it's not the same figures that you're going to see in the paper. It's going to be a little bit more fun. So thank you for the introduction. And that was like all of my bits of paper and stuff. But, I'm really, neck deep in the cannabis industry, both sides, the hemp side and the THC type cannabis side. So, this is my passion. This is what I do. So the aroma of cannabis, this is a hot topic right now. It's all about the terps, right? So, this, although I haven't talked about this much, I think that this experiment was one of the coolest things that I got to do during my PhD research. Well, it's all cool, but this one hasn't garnered as much attention as the other ones have, the other experiments and the other publications I have. And while I'm going through this, like something to keep in mind is that all of this work was really grassroots. I didn't have any big funding source. It was all like little tiny internal grants, $400 here, $200 there. So all of this cannabis that I had to purchase for this, like a lot of it came out of my pocket or from these small grants. So, some of these other big studies that you're seeing with, hundreds of samples, like I just didn't have the resources to do that. But even though that's the case, the results are pretty cool and definitely give a proof of concept to further more research opportunities in this direction. So here we go, and let's look at the genetic versus aromatic consistency in cannabis. So first of all, just a quick overview of what I'll be walking through today. First, I'll go do a basic overview for which a lot of people is going to be old hat, but I want to make sure that everybody who sees this is up to speed. And so we're all on the same page when we start getting into the science. I'll give you a little bit about what I did during my PhD, the genetic tools I used, and kind of how this stuff all fits together. Then I'll talk about the acquisition and the genetic analysis of the samples that went into the sensory analysis. Then the sensory experiment, how it was conducted, how the data was gathered, and then how I analyzed it. And then a hard look at the compounds that were driving the sensory perception. So the terpenes, obviously. And while as I said, all of this is in my published paper and in my dissertation, but the way that I'm going to present it today is going to be a little bit different. So the information that you get from this talk, you won't see anywhere else. So you are getting a first look. And then after that, I'll wrap it all up with some implications and the conclusions. And then hopefully we'll have a good portion of time to have some Q&A. So first off, some basics on cannabis. The plant makes cannabinoids and terpenes along with a whole slew of other compounds. But generally, the terpenes and the cannabinoids of what people have historically been interested in. The cannabinoids lend to the effects, while the terpenes are responsible for the most part, or at least we think, to the flavors and the smells. and they might also help with some of the effects. But there isn't really much research on terpenes pre and post combustion, because most people smoke cannabis, like what actually reaches your bloodstream and what that translates into. So, but anyway, I digress. That's not what this is about. Just wanted to, let you know where I'm coming from in that. So we have all these compounds. We have terpenes that are ubiquitous in the plant world. All the smelly plants have terpenes, and they're thought to have several functions in the plant, including as an insect repellent and communication with other plants around. So, like, hey, there's something eating me. All of my friends, you guys need to upregulate your defenses, or you're going to get eaten too. So, I like to say that the smell of fresh cut grass is actually the smell of your grass screaming, and that's horrifying. But that's what terpenes are. So, for example, humulene is found in hops, linalool is abundant in lavender, pinene, obviously, in pine needles, myrcene is found in mangoes, limonene is in lemons, and so on and so forth. And then we have the cannabinoids. THC and CBD have gotten most of the attention over the years, but there's many others. There's over 100. Some pack much heavier punches than others. We're just learning about this with THCP, for example, which is said to be about 30 times more potent or stronger than THC. So we've got all these things working together in some capacity in the brain and the body to produce an overall effect. But it also depends on the combination and the level of these compounds that lead to the effect. So while we have these 6 terpenes in combination with 6 cannabinoids, they can have very different effects depending on how much of each is present in the material. And if you kind of try to wrap your head around this, you can imagine the millions, the limitless combinations. And this is only 6 of each. So, like, thinking about this, just like my brain's going to explode. And especially when we know so little about the vast majority of these, we really don't know much about this. And looking at this slide, I think you can kind of get the message. But I also I like to think about cake a lot, for, because cake is good. But so I equate this to, like, in a cake, you've got, or any baked good, really, you've got flour, sugar, water, eggs, the common ingredients. Those are important to make the cake, right? You can't have a cake without some of those things most of the time. And then you've got flavors, things like chocolate, vanilla, fruit, and those kind of shape the flavor of the cake. But it's not really those things that make something incredible or different or give you the, unique experience that you have from different kinds of, vanilla cakes, let's say. It's the small nuanced ingredients that can really make a big difference. So salt, for example, if you put a lot of salt in a cake or a little too much, it's going to be disgusting. Nutmeg, clove, cinnamon, maybe cream cheese or something, sour cream or something a little bit different, maybe a little applesauce to make it, have a different texture. The things we don't normally think about unless we are like, hey, this is amazing, what's in it? So that's kind of how my brain thinks about it. And so hopefully you can get on board with that kind of thinking as we go through this. And another thing to think about is that in terms of terpenes and some of these other flavor/aroma compounds, 1 + 1 doesn't equal 2. 2 of the same terpenes in the same ratios may be completely different when you add a little bit of something else, like a little bit of limonene or even a lot of limonene. It can completely change everything. And that's, all these combinations just can be completely different, even when they're slightly similar, but not exactly similar. And the fragrance industry knows a lot about this. And it's one of the reasons that some perfumes are so hard to recreate. Chanel No. 5 is a really good example of something that is really not recreatable because it's got something in there that they just can't get their finger on. So something is missing at a minute level, but it makes a huge difference. And, we're learning that some of the thiols in cannabis, for example, are detectable in the parts per billion. Very, very strong in very, very small quantities. So what I'm getting at here is that the terpene profile, especially when you take a handful of the most abundant terpenes in a sample, isn't really going to tell you a lot. there's so much more to the equation that we don't know, and we don't know the weight that each holds in the different combinations. So when we come up with like new strains, for example, so this purple punch in the top row, we don't, although we have these major, these most abundant terpenes, it's not going to be limonene is energizing and caryophyllene is calming and pinene gives you an alert, because that would be really a weird experience. But really what you have is you have the environment, the cultivation, the processing, they're all going to shape sort of a combination of these things, which will then lend to an end product. And so really, this is the cornerstone of all of my research. Genotype, so your starting material, plus the environment, and this is all the external inputs, including the growing conditions, obviously, but also harvest time, so the maturity of the flower when you cut it down, the curing process, so it doesn't end at the cut, it keeps going. Storing, how did you store it? Did you put it in an airtight, dark container, or did you put it in a clear jar on a windowsill with no lid? And then the time to sale, so, or time to consumption even. So how long was it sitting around? Those are all going to feed into the final product, which I call the phenotype. So whether you're calling the phenotype what it looks like at harvest, or whether you call it the chemotype on your COA, this is all part of the phenotype. And if you alter anything on this left side of the equation, the right side will also change. So as I said, like, think about storage time, how that product changes, and that, even when you purchase something, you have a COA, think you know what's in it. It's been probably sitting around, maybe not for very long. Let's hope not for very long. But then you take it home, you put it in a drawer, you wait a couple of weeks. Like, it is not the same product as what you have on the COA. That COA is just a snapshot in time, really, of what made it into the lab. It's not really exactly representative of what you have in your hot little hand or in your pipe or what gets to your lungs. And so now we've arrived at the University of Northern Colorado in 2015, when I began my journey looking into the relationships of cannabis using genetic tools. So over the years, I had a ton of different ideas. These are the ones that made it to publication or to fruition. These are the ones I was allowed to do. These are the ones that didn't get reined in because I am very ambitious. And most of this has been published, so you can read it on your own. But basically, we have the variation within strain experiment. We've got the looking at the National Institute of Drug Abuse, like, what is it? We've got the smell, the aroma experiment, which I'll be talking about today. Phytochemical variation. The THC inflation just came out last year, which you're probably familiar with. And then I have another cloning experiment, which is still ongoing because I didn't finish it before I graduated. But that's looking at genetic changes from the cloning process. And so, like I said, these are the three I'm focusing on today: genetic similarities and differences within the same strain, the sensory perception through organoleptic methods, basically sniffing weed and reporting smells, and then looking at the actual chemicals of the terpenes in those samples. And so this is like a quick visualization of my genetic methods. Don't worry if you don't get it, it's okay. But basically, I bought a lot of weed from dispensaries and then looked to see if the flower labeled with the same name had the same genetic profile. So these chromatograms right here, I don't know, you can see my cursor, I hope. These are all Purple Kush purchased from different dispensaries, different cities. They should all be exactly the same. If all of that flower is, from clone or from seed, they should be very similar. And just looking at this right away, you can tell they are not the same, right? So puzzle solved, game over, I graduated. Not really. I wanted to do a lot more work and needed a lot more samples to give it, academic and scientific robustness. So this was, my first published work in cannabis was Genetic Tools Weed Out Misconceptions of Strain Reliability, Implications for a Budding Industry. And yes, I know there's a lot of puns in there, and I was so excited to do it. But yeah, anyway, so this was just looking at strains with the same name. are they genetically similar, identical, or not at all? And you can really easily see, even though you don't know what it is, these blue dreams right here, they should all be the same. Or actually, this is Durban poison up here, sorry. All these Durban poisons right here that were bought in different cities, they should all be the same. So they should all be, look the same in this graph. And you can clearly see, like, some of them are, but this one's not at all the same. And so I went through these 30 different strains that were purchased in 10 different cities, 3 different states. And, the take home message here is that sometimes there are genetic similarities like you would expect to see, but in 27 of 30 strains, there was at least one serious genetic outlier. So this blue dream here, not at all similar to the other ones. And yeah, so we do have genetic differences where we wouldn't expect to see any. But what Twinkle brought me here to talk about today is my sniff study. Can people sniff out a genetic imposter? So this has to be a 2-part experiment. So first we need to go around and find some genetic outliers, and then we need to follow that up with a sensory analysis. I had a lot of genetic data, but for the sensory portion, we needed to have enough flower for these panelists to smell. So what I needed to do was figure out what I had good data for, what was popular, what we would be able to find, to be able to pull all these things together and come up with enough samples to pull into the sensory analysis. And I partnered with Dr. Avery Gilbert on this, who is an aroma expert. He's awesome. And he actually has worked with Twinkle and Abstracts on their terpene research. So we have a connection there. And so the genetic portion, we kind of looked at a graph like this earlier. As I said, we had some extra genetic data. We went around, I purchased a bunch of flower. There were 5 strains that we had. And you can see, let me backtrack a little bit. You can see down here that these are the sample names and they don't really mean anything. These are all OG Kush. I've got SN and GN. So GN just means that it was we had enough to do a genetic analysis on that sample, but we didn't have enough for somebody to pull it into the sensory portion of the analysis. So we needed enough genetic data to show that something was completely different, so we could pull that in, and we needed enough material to make sure that we could use it on the sensory portion. So for example, this OG Kush, we've got 1, 2, 3, 4, 5, 6, 7 samples that I went out and I bought 2 grams of, and I added this one extra sample that I had enough just to run it as another sample to boost up the genetic, to show that it was genetically different or something was genetically different. And you can see, like, all of these are really, really similar. That's what we would expect to see. That's lovely data, but none of those are outliers. But we did get one that happened to be completely different. So that's perfect. Love that. That's what we wanted. That's what we needed. Same thing for Blue Dream. We've got a lot of sample from here, but this 7 and this 2, like, they're exactly the same as 4, 5, 6. So we use some of those, and then we, of course, have that genetic outlier. In Durban poison, we didn't have a clear genetic outlier. Like, this one was different, clearly, but it was we only had a little bit of it. We didn't have enough to use for the sensory portion. So, but what we did have was these 3 samples that were genetically, like, completely identical, no difference at all, not one letter out of place, nothing. And so we used that, those Durban poisons as kind of our control for the experiment. And then Mob Boss, we had 2 that were genetically identical and then one that was different. So that's what we had to work with. And so then when I had figured out, okay, so we've got these samples, these are all genetically identical, these samples are the outliers, we blinded them. We put them into glass jars with a number identifier, and I had that information, and then I passed it off to Avery, and he didn't have any information. He just had glass jars with numbers, and he conducted the sensory portion of this study. And so we had 15 samples, and that doesn't sound like a lot, and it isn't. However, people have a threshold of how much they can sniff before their nose kind of gives up and doesn't really pick up on nuances anymore, and the data kind of becomes useless. So the threshold on that, according to Avery, is 15 samples. That's about as much as the human nose can, keep telling different smells apart. So that's why we topped out at 15. We had 40 odor descriptors, and you can see all of them here. We had 55 participants. It was a reasonably even mix of males and females. And we just put a call out on social media. And so they weren't expert sniffers. But that's kind of the point. Like, this wasn't for experts. This was for you and me. What can we smell? Can we sniff out a genetic imposter? So that was perfect. And it's not like any of these smells, we wouldn't know. Like, what butter smells like, what something sweet smells like. So we thought that was okay. And according to Avery, this is how it's done. So he's the expert on this. So we just went with it. And they reported what they could smell. And so these are my groups. And I don't know if this is in the way. It might be in the way. So this was a lot of data. So we've got 40 descriptors, 55 people. And if you can imagine a matrix with all that, it's a lot of data. So I needed to kind of boil it down to something more manageable and meaningful. So I took the odors and I kind of put them into groups that I thought made sense. And so the groups that I came up with were pungent smells. So things like ammonia, skunk, gas, blue cheese. They all went into the pungent group. Sweet were things like fruits and candy and honey. Floral was fresh cut flowers. So rose, I don't remember any of the other flowers, but floral smells, floweries, lavender. Spicy was things like, I got these mixed up. So spicy would be like the dried spices, pepper, I don't remember, pepper and stuff like that. And then earthy would be dirt, coffee, nuts, butter, things like that. And what I wanted to see is, what was there big differences between those consensus samples, so the ones within a strain that were the same genetically, and the one that was completely different. And so in the genetic identicals here, we have multiple samples, and the outlier is just that one that's completely different. And so with this, you can see, I'm gonna hopefully add a box. Okay, so you can see that in Blue Dream, the outlier was much more pungent than those genetic consensus samples. In Ma Boss, the outlier was more sweet and floral. In OG Kush, the outlier was more pungent and more earthy. And other differences, in Ma Boss, the outlier was less pungent, and in OG Kush, the outlier was less sweet. So there was significant differences or substantial differences in the outlier compared to the consensus samples. And even when you can see like the error bars, like this is way more floral than anything that was in the consensus sample. So they were different. But then I was like, but I don't know that much about aroma science. Are these real groups, or are they just things that, I thought smelled the same? And I don't know, like, I don't know. So I wanted to see if there was something out there that maybe somebody in the cannabis industry had come up with in terms of, these aromas and what cannabis has to offer. And so I came across this. There really wasn't anything out there at the time. This was a few years ago now. This is a wheel from the Western cultured terp wheel is what it is. And I know you can't see, like, the writing's very small, but almost all of the aromas that we had in our study are represented somewhere on this wheel. And so I thought this would probably be a pretty good set of groups to put our samples into. And so spicy, sweet, earthy, and sour. And when we looked at that, and I didn't put boxes on this, but you can see the outlier in Blue Dream, for example, was less sweet and more bitter. In Mob Boss, the outlier was much more sweet than the consensus samples. And in OG Kush, sweet, sour, spicy, those are all pretty spot on, but the outlier was less bitter. So we are kind of, again, seeing that the outlier was different from those consensus samples. So the answer to the question, can people sniff out a genetic imposter, is yes, but and so moving on to, this is the stuff that isn't in my dissertation. Well, kind of is, but this is a little bit different. So I wanted to look at the pooled samples. So pooled top 5 aromas, and I only have, these are only consensus samples. So we're not even looking at the outlier anymore. We're just looking at things that should be the same. So we've got 3 Durban poison samples here. And remember, we've got three samples. So the top 5 aromas for each one of the samples. So that's why there's more than 5 aromas here. But you can see the difference. And this is the number of people that reported this scent. So for this sample, 23 people reported that this gray sample, whatever sample that was, 4SN, was flowery. In this sample, only 19 people reported that this sample was flowery, and on and on and on. So for sample 1, it kind of had this aroma profile. Sample 2 had more of like a more flowery, a little bit less fruity. There's no earthiness to it. And then the 3rd sample had a woody scent. It was a tea was thrown in there. So you can see that even though these are genetically identical, they still have very different aroma profiles. So we've got a lot of variation even when we have the same thing. And so on. So here we've got the mob bosses that were genetically identical. One of them was much more earthy than the other one. OG Kush, we've got lots of variation here. We've got tea and not so much tea. We've got this sample that was much more chemical. This sample was a lot less pungent. And Blue Dream had a lot of variation in it too. Not only like among the top 5 pooled, but like there's so many more flavors, aromas in Blue Dream that people perceived. So it kind of got a little crazy, but you can, I mean, I think I'm making my point here is that there's a lot of variation even when things are genetically the same. And so here is just sort of the scent profiles for the different strains, because I was like, well, maybe they're just all kind of the same smelling strains, but they're not. Like you can see Durban Poison and OG Kush have this very different floral smell. So if we go on, I thought, well, what about the actual terpenes? So we did some terpene measurements, and I have some graphs coming up, but we measured 21 terpenes. And I just picked the top, what is it, 6? And put them in a little pie chart to show, okay, what are we looking at in terms of terpenes? And so they're all beta caryophyllene heavy, right? That's the most abundant terpene in all of these strains. Okay. So then I thought, well, maybe beta caryophyllene doesn't matter. Like maybe it doesn't smell that much. Maybe there are other terpenes that are like taking over when beta caryophyllene is the most abundant terpene. So then we break that down, ignoring the beta caryophyllene. We've also got in mob boss, we've got nerolidol 2, we've got alpha pinene, and alpha caryophyllene is what the analyzers gave me, but that's humulene. And then we compare these raw terpene profiles to, whoops, I go backwards. These actual terpene results to the scents, like this, the groups of scents. We've got mob boss being pungent, earthy, and spicy. Do these terpenes translate into that? And I don't know, like it doesn't look very floral. So I'm like really confused by this. But as I said, 1 + 1 doesn't equal 2. If we've got, something that's woody and spicy and we add herbal, woody, fruity citrus, we don't get that same scent profile that you would expect. It turns into something different, something nuanced, and it becomes very difficult to try and figure out what exactly is what. And so Blue Dream, same thing. We've got that beta caryophyllene that was predominant. And then the next terpenes were alpha pinene, myrcene, and alpha caryophyllene. What we smelled in the Blue Dream was earthy and spicy. I don't know, there's no like I just, it does to me, I don't know. I haven't really dived into this, but I just wanted to kind of put it up there. Like, it doesn't translate well to me. So here we've got floral, and we've got 2 floral scents being the second terpene, most dominant terpenes, but it's not a very floral profile, which is interesting. We've got sweet way up there, and what here is sweet? I don't know. So I'm thinking, maybe it's not the terpene profiles that we want to be looking at, because they don't seem to really translate well. We do have a lot of floral in the Durban poison, and this floral, piney, herbaceous. There's 3 flowers, so that one makes sense to me, which is nice. So maybe maybe there's a hint. I don't know. This is all really, complicated stuff. And so looking at the actual terpenes that we measured in these strains, we've got Durban poison, again, all genetically identical. And you can see there's some serious variation in some of these terpenes. OG Kush, the outlier is in green, so that should be the weird one, but it's not in all the cases, right? So same thing with Mob Boss, the outlier is in green. And again, it's not always the one that's out of place. Blue Dream, the outlier is in green again. It has less variation in each terpene, but a lot of variation in the actual smells. So then I was like, well, what about Leafly? Like, Leafly gives you terpene profiles, like it's heavy in myrcene or it's limonene dominant, whatever. So I was like, okay, let's see what Leafly has to say. And so Durban poison, they report, has terpinolene as the dominant terpene, which gives it kind of a fruity smell. So here is our terpinolene. Sure, one of those samples is definitely dominant in terpinolene, but this one, and these again are all the ones that are genetically identical. I didn't put in the outlier, but this first sample here in green, it's not dominant in terpinolene. And then the next one is myrcene, but we have caryophyllene over here. It's got more caryophyllene than myrcene. All of them did. So OG Kush, Leafly says the dominant terpene is myrcene. Again, we've got beta caryophyllene as the dominant one in most of them, except for this orange one. But, really what I want you to take home from this is my little tiny, samples of 3 or 2 from each strain, that's a very small sample size, but none of them match what Leafly has. Leafly's in California. I'm in Colorado. Maybe that has something to do with it. Because again, the whole crux of my research is that genotype plus environment is what leads to the phenotype. What they're growing in LA and other places in California may not be anything like what we're growing in Colorado, whether it's inside or outside. And because completely different states, different regulations, different methods that have to be done in terms of like what can you spray on the plants and how do you sample and doing all these things. All that is going to feed into these differences, right? So we can kind of assume that all of these differences are coming from external factors, especially when we know that the genotype is the same. And I can guarantee you, and I've known this forever, that if I gave Kevin Jodry a clone and I took home a clone and we both grew it, they're going to be completely different at the end of the day. And it's partly because he's a good grower and I'm not. But it's also because he lives in a completely different environment than me. We'll probably have completely different growing styles. We'll use different media. We'll use different nutrients. We might have different lights. All of that is going to be different. So, and that's the premise behind the grow off, right? Is take this clone, go home and grow it, and we'll see who's the best grower. And, I also think about bananas, right? So you can take a banana, depending on where you store it, it's gonna age faster. It's going to have a different flavor. It's going to have a different texture. Even if it's the same banana over time or bananas from the same tree, but different bananas. That's, kind of how I think about cannabis. It's not that different. Or, back to my baked goods. I love baked goods. You can start with the same ingredients, but subtle differences in the ingredients, the temperature of the butter, whether you melt the chocolate or not, the kind of oven you're using, the tray you put the cookies on. Those are all going to create little tiny differences that really make a big difference in how your cookie comes out. Again, this is the premise behind Nailed It. Like, you can start with everything the same. You can have the same recipe. You can have the same directions, but it's not enough to have the same genotype to have everything come out the same. External influences are really super important. And I see this even in our grow. Like, all of these are the same strain from the same company. We have 5 greenhouses. They're all a little bit different in their own special ways. Some beds are up off the floor. Some beds are on the ground. Some beds we have more plants than others. So we get all of these variations in our plants, even though they're all genetically, very similar. They're all the same seeds. So really, it's crazy to me how we come up with all these different things when they should all be the same. And so, some implications of this would be, if you start with the exact same, genetic material as we do with this Durban poison, like I did in my experiment, they were all genetically identical. You grow them out. You might have slightly different conditions. Even if you're all in Colorado, every grower has their different methods. There's different, growing styles. So hydroponics, aquaponics, soil, whatever the case may be. You're going to end up with slightly different flowers at the end of the day, with slightly different chemicals and different variations, which to the end consumer are going to be perceived differently, whether that's aroma or effect. But yeah, so slightly different effects. They're all happy, but just a little different. But if you start out with different material, even if it's just slightly different, you grow them out, the difference are going to start to compound. Your differences in your plants are going to be measurable. Your dried flower are going to be very different, and the effects on the consumer, the smells and the effects will be even more different. So it's really important that you start with something that you know what it is, and then you can tinker around and create differences. Because even though it's Durban poison, that starting material is different. You could end up with very different things. And everybody I know that consumes has had this experience where they've had a strain that they thought they knew what it was, and it did something completely wacky to them, and they're like, what the hell was that? So this is all going on. And that's sort of the end of my soapbox rant. But the conclusions, your nose knows, you can sniff out an imposter, but there is, a lot of variation within strains. So even when you are smelling, Chem Dog, there's going to be some variation in there. But if there is something completely different, you can tell. Generally, you can tell. So if something's not quite right and you're not feeling it, even though it has a name on it that you're familiar with, it's very likely that it's not the thing. If we wanted to get to a point where we wanted to have consistency, so this would be for like maybe medical applications. In order to get consistent products, there should be genotyping. So you got to know what you're starting with. Make sure that, your Chem Dog is Chem Dog. And then growing standards. So if you grow like this, then you will get this product. If you deviate from those standards, you're going to get something completely different. And then consumer education. So most importantly, we need to let the consumers know that in the cannabis industry, Skittles may not equal Skittles. In the other consumer world, Snickers is a Snickers no matter where you get it from. That's not necessarily the case in the cannabis industry. And I know there has been some research that has concluded that genetics plays a larger role in variation than environmental conditions. And I have to disagree with that, because my research shows that you can start with the exact same genotype, but if you grow it under different conditions in different, these were all from different dispensaries, different grows, different lot numbers, different batch numbers, different license numbers. We know that these were grown under different conditions, and they came out very different. And then just one last thing, I know there was probably some people cringing. saying, oh my God, she's saying strain. Oh my God, she's saying strain. It's not a strain, it's a cultivar. I'm just going to say strain in botany refers to a group of descendants from a common ancestor that share a uniform morphological or physiological character. It's a perfectly fine word to use for anything botanical. There are strains of rice and wheat. It's not just reserved for bacteria and viruses, as many people proclaim. It just isn't a taxonomic rank. So you can't have like, you couldn't enter a strain into a competition that's reserved for cultivars, like cultivars of tulips. They get names and they get registered, and they're, you know, very consistent, uniform, unique. We don't have that in cannabis. So I say strain because cultivar is like elevated from that, which most cannabis doesn't reach that threshold, at least for me. So I say strain. It's not a bad word. So I just wanted to put that out there. And I guess with that, I'll say thank you for paying attention to me drone on and on and on about this. And if anybody has any questions, and if we have time, I'd be happy to entertain them.
I think that was great. We do have time for some questions. I have some questions. Thank you so much, Dr. Schwab. That was a really awesome presentation. I've read your paper a bunch of times, and so it was really cool to hear all these different aspects of it that I haven't got to see, and like also just get a glimpse into your personality and what your life was like as a researcher before, which I think is really cool. And something that I want to talk about more about in our journal club, which is like you mentioned in the beginning, like the grassroots funding of this research and how you had to work really hard to make it like up to the academic and scientific robustness standards to be able to publish this data. And I
Yeah.
think that's like a really big challenge in the cannabis industry with the way that we have to do research and the lack of funding, especially within academia. And
Mhm.
it's not something that a lot of people know about. And I just thought that was like as I'm learning about this in the industry now, it's really awesome to hear that you've also experienced these same restrictions and having to overcome them, and how you made this a two-part study. And I have some questions about that, which is when you first started out with this project, did you initially just start with the idea of doing genotyping, and then after seeing the results you were like, oh, we have to tie the sensory into this, we have to tie the terpenes into this? Or did you know from the beginning that this was going to be like a multifaceted research project?
So the way this all went down is that I published that first paper, Genetic Tools Weed Out Misconceptions, blah, blah, blah. Avery contacted me and he's like, hey, I'm a sensory scientist and I would love to meet with you. And we met over a beer. And as soon as we sat down and he started talking about, you know, terpenes and sensory experience, I was like, I totally know where you're going with this. Let's do it. And so he wanted to know, like, can people sniff out a genetic imposter? He's like, you know how to do the genetics. I know how to do the sensory portion. Let's get together and make this work. And I was like, oh, hell yeah. So that's how that went down.
That is so cool. That is really, really cool to see two different branches of scientists coming together to make a brand new, because it is kind of a brand new thing to have not there's not a lot of published
It
science
was
data.
it was really hard to get published because in a genetic journal, they're like, what is this sensory woo-woo science? And in like, where do you put the sensory stuff? They're like, we don't know anything about genetics. Like, we're a perfume, you know, we deal with perfumes and aromatic oils and things. Like, what is this? And so it was really hard to find somebody who was willing to make that leap into that bridging of two different, completely different fields. I think it was published in a psychology,
Yeah.
cognitive, but it was part of Frontiers, right? So it was still under a very legitimate journal umbrella, but it was in a specialized, like, very tiny area of their journal. So that took a lot of work. It took us a long time to get published, actually. Yeah.
Wow. Did your first publication, the genetic differences, was that published in like a straight genetics journal?
Actually, it was the first publication in the Journal of Cannabis. Is it Cannabis Science? Well, the journal that CSU started.
Oh,
Um,
okay.
so I I presented that information at their first conference, which was awesome because Daniela Vergara also presented at that conference, and we were kind of doing two, like the same kind of thing, but she was using genomics and I was using like old school genetic tools. And we kind of came up with, like we're not getting different results. So we were like, okay, that's pretty awesome. And yeah, so then people also don't know that you have to pay to publish. You don't get paid. I don't get paid. It's usually a couple thousand, $3,000-$4,000 to publish research. But this brand new journal was like, if you publish with us, if we, we'll peer review, if you publish with us, we'll waive the fee. And so we were like, oh, perfect, because we don't have any money. And so, and it was like one of the first cannabis journals, and so it wasn't that bad. So yeah, so that's the reason that we published there, was we didn't have to pay.
That's awesome. Yeah, and I love that this paper is open access also, which it kind of feels like, I'm sure you had to pay more to make it open access, but it's really nice, especially in cannabis science, when people can access them without having to have academic affiliations or pay to read them.
You can
Uh,
just email the authors. Usually they'll give it to you.
yes, totally.
Just everybody who's listening.
And then I actually wanted to comment also on, let me see if we have any questions in the chat. If you guys have any questions or comments, please do feel free to put them in the chat. I love the slide that you had with the wheels, the aroma wheel, because I thought that was really cool. I thought you were going to be going towards genetic strain validation, and you totally went in a different direction than that, which I thought was really, really interesting. I like how you used the sensory data that you had with the wheel as an afterthought to be like, okay, does this make sense? And are we capturing what this strain or cultivar actually smells like? And I thought we were going to come to a decision where you were going to be like, and this is what Mob Boss should look like. But you didn't. And I actually love that, because you're right. I hadn't thought too much about all the differences that would, like all the possible environmental differences that could change something. And you made a lot of different small nods to that I hope people picked up on, like the packaging for the bananas, because packaging with cannabis is also so important. Like the time from harvesting to getting it in your hands. And like you said, a snapshot of that is really all we get when it's in the lab, not a complete profile or an accurate
And
representation.
you can purchase like the most perfect bananas at the store. Like they're just the perfect ripeness, the beautiful, they're all yellow. You take them home, you forget about them for 2 days, and they're trash. Or you put them in the drawer in the freezer, or like in the fridge by accident, they're black, like all parts of the handling process for cannabis matter. If you want to keep your, really perfect, expensive, quality cannabis, like don't leave it sitting out on your desk where there's light shine, like you can screw it up just like you can screw up a banana.
Yeah, or baking a cake, which is much easier to screw that up, I feel like.
But some people are like innately good at it. It doesn't matter. You give them a handful of wood chips and they'll make it into a cake. But some people, you can give them the most detailed instructions. You have to do all of these steps in order to get this, and they screw it up every time. And it's not. It's not their fault. Just some people are not good at baking.
Yeah. And some people don't have a green thumb, and that is real also. I do. I also I'm pretty
Look at my
okay.
bananas.
Yeah, that was great. And then I think that was it. I learned so much from that. So thank you so much for sharing with that. And then another thing that you had mentioned was that slide with the mixer. Somebody else said they really liked that slide with the cannabinoids and the
Dialed
terpenes.
in, like
Yes. But like that's only part of it too. Like when we actually think about it, it's so much more complex than that. There's way more than 6 cannabinoids, there's way more than 6 terpenes. And we're only really looking at the before product. We're not even looking at the inhalation product, the combustion products that we're creating, like thousands of new molecules as you're consuming them or as your liver is digesting them if you're eating them. Like,
yeah, and like a lot of these compounds are volatile. You put a flame to them and they're gone.
yeah.
Like are these limonene heavy dominant uh cannabis or is that even making it into your system? I'm guessing not. So does the energizing research that says limonene is energizing in a, bath wash or a lotion, does that translate to cannabis? I don't know. I'm not convinced, like I need to see some research on it. So I'm I don't know.
Yeah. And
Yeah.
just from personal experiences and talking with other people in the industry, there doesn't seem to be I feel like there's two groups of people. There's people who really feel that those effects are true to what they experience, and then there's a group who it doesn't matter what they consume, their effects are going to be different every time. So, I feel like it's hard to know is what causes people to be into those two different groups, and maybe it really matters for some people. Maybe they will feel that energizing effect from limonene, and the other half of the population never feels it at all.
Yeah. And I feel like that's the same with bath and body wash. You tell somebody that it's lemon scented and therefore it's energizing, and they're like, yeah, totally. I use it in the morning and I just get this great, burst of energy. And it's like, well, is it that, or is it because we told you that's what it's going to do? I don't
Yeah,
know.
and that's the weird thing about sensory science in general and smell science in
Mhm.
general, is that there's so much like personal um bias involved in it that it's really hard to. That's why I think sensory panels are so interesting and cool, especially like a 55-person sensory panel is a large panel, and that's hard to pull off. So that's really interesting that you were able to find trends within that big of a group of people, because
And just so everybody knows, that sensory panel, it was conducted in like a white room. There was a, white in front of them, brown bottles, just a number. There was no music playing. There was nothing to influence anything that the person was experiencing. And like, I've talked to some people about doing like a smoke study, and I'm like, you can't have people like bring it home and smoke it, because they're going to go into their bedroom or their living room, turn on some music, put the TV on, like drink, a glass of water, I don't know, like what, pet their dog, like all of that is feeding into your experience. So it's really not like a controlled study.
yeah, and it's like someone might be having the best day and someone else might be having the worst day, and they're both using the same product, and they're going to have two different experiences because of how they felt when they went into the scenario.
Or what they ate, or how tired they were, or like so many different things. Yeah.
Yeah, so you need a really big sample size to be able to try to get some information from that.
Yeah.
Um, Aaron Appleby says, "What do you mean by volatile compounds are gone after heated?" So, do you want to answer or I can answer?
You're the chemist. I have an understanding of what it is, but you could probably explain it better.
Volatile compounds like thiols or other terpenes, volatile just means that they have the ability to, let me just think about how to put this. Um, it's so funny when I write about this all day long and I can't in the moment think about it. But basically, they're volatile, so they're like not evaporating, but they're being removed from the product just by existing. The environment that is around the product is where they want to go instead of staying in your product. So when you break open a bud, if it has a lot of thiols in it, you smell the thiols right away when you're breaking it, and that is them volatilizing out of the product and reaching your nose. So they're so after you're heating them, so if they're already ready to like evaporate from the product in the regular environments, like just when you break it open or in room temperature when there's light around, heating is a pretty serious chemical reaction. So there's a lot of energy going on there, and that energy is going to cause them to volatilize faster, and so they're going to be like leaving your product faster or being converted into combustion products faster than some of your other non-volatile compounds. And so non-volatile compounds are things like THC and CBD. They're larger molecules, they're heavier, and they're more prone to stay where they are in the solution already. I hope that answered with enough depth and understanding. And we are almost at time here. Lauren says, "Thanks for the discussion and awesome journal club." I have to agree. Thank you, Dr. Schwabe, for joining us.
Thank you. This was so fun. And like I said, I haven't talked about this for such a long time, and like making these new slides and talking about it from a different sort of lens, like very cool. Thank you so much for having me. I really enjoyed it.
Yes, and I have to say, like, the reason that I was so excited to ask for you to come is because I'm writing a paper, and I've been referencing your paper a ton. And so this was my personal bias on Journal Club, where I was like, if Dr. Schwabe wants to come talk about her paper, I would love to hear about it. And I'm glad other people enjoyed it too.
Yeah, so fun.
Okay, great. I think we can tune out a little bit early today. And if anybody wants to see this video, it will be up on our YouTube channel, and we'll be posting some clips on it also. Okay.
Wonderful.
Thank you, everybody.
Thanks, guys. Bye.